Welcome to this free webinar on genomic data analysis. This webinar is part of a special 6-month program developed in collaboration with Noguchi Memorial Institute for Medical Research in Ghana. Omics Logic programs are a result of close collaboration between Pine Biotech and innovative big data.
Welcome to this free webinar on
I apologize for the interruption. Let me start by sharing my screen again. So as I was talking to you about the Omics Logic programs, they are part of a result of close collaboration between Pine Biotech and numerous academic institutions around the world who have participated in content evaluation, curriculum review, and project design. As a result, the Omics Logic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. Our goal is to prepare biologists and clinicians for the influx of data, associated methodologies, and data-driven research projects as they are prominent with the emergence of computers for the tech industry. More and more biologists and clinicians are now expected to understand and handle their own data to publish and succeed in basic and translational research. In fact, biology itself is rapidly acquiring the character of a data science. So this is what we hope to achieve through these Omics Logic programs. So the first of this 6 months program was the bioinformatics for infectious diseases, focused on the use of bioinformatics and big data for infectious disease research, diagnostics, as well as drug and vaccine development. This program is going to be ending on March 10th. One of the major focuses of this program was to support ongoing research projects, which are spearheaded by Dr. Anita Gansha and Dr. Bright Adu's group at Noguchi, and her collaborators who have a long history of research in Ghana and other countries in sub-Saharan Africa to track, monitor, diagnose, and study the spread of drug-resistant malaria, as well as identify novel approaches for vaccine design, as well as the interaction between malaria host, vector, and pathogen to help conduct such research. The upcoming Omics Logic Genomic Data Analysis program is the second phase of the 6-month-long program. In this period of training, participants will get an in-depth overview of concepts and analytical tools used to study genomic variations in infectious diseases. Each week, participants will meet online to discuss various aspects of NGS data processing, genomic data analysis, and interpretation. The program starts on March 12th, and today we are going to be talking about this program at length. In the genomics program, one of the major focus will be on the application of bioinformatics to malaria-related research, where we will be looking at the efficacy of vaccines and drugs associated with resistance and the complexity of host pathogen vector interaction. For this purpose, our team has already prepared a number of research data repositories and databases that will help the participants identify high impact data sets for analysis. You can see some of them published in our recent blogs. So Dr. Mohit, if you can kindly share the blog with everyone in the chat. We will also discuss various applications of genomics to the study of SARS-CoV-2, where selective pressure on various proteins will lead to the emergence of new variants. Such changes are already being seen as a result of increased immunity, vaccination, and other host pathogen dynamics. In this upcoming Omics Logic program, you will have the opportunity to interact and collaborate with our international team of mentors. Let me introduce you to them. We have Dr. Harpreet Kaur, who is a certified Omics Logic trainer and expert bioinformatician. She will guide the hands-on sessions and assist participants with research projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is our Omics Logic project mentor and an expert in big data computational analysis. He has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is the CEO and co-founder of Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners who are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will now invite Ilya to speak more about genomic data analysis. Over to you, Ilya. Thank you.
Thank you very much, Pipsa, and welcome everyone to this webinar. Great to see you. As we get started, I'll talk a little bit about the genomic data and what kinds of research we're planning to do in this program. But I also wanted to get to know a little bit more some of the participants today. So if you can, please share maybe which country you're coming from to this webinar. And also, if you can share the name of your university so that we have a list of contacts that are here today. Let me just check on the chat. All right, great. So we have somebody from Italy, from India, University of Delhi, the Philippines, Pakistan, Iran, India. All right, University of Karachi, Chile, India. All right, Paraguay, University of Management and Technology, Cairo University, Lucknow, India, China. All right, Canada, Egypt, Bolivia, Nigeria. All right, excellent. So a lot of people, Tanzania, Kenya. All right, excellent. So welcome everyone, Benin. Okay, welcome everyone to this webinar. I hope that you enjoyed today's presentation and that you will be able to join us for the extensive training program that we will have starting in March. So in the last 20 years, availability of genomic data has led to an increasing understanding of how cells function at the molecular level and has allowed us to understand some of the major causes of deadly diseases. And with the current rise in viral outbreaks, persistent bacterial and parasitic diseases, and their persisting deadliness, there's a major focus on developing tools, data repositories, and analysis methods for a better understanding of the reasons why these diseases emerge, how we can treat them, and how can they be potentially prevented with measures like vaccines. And so as a result, there has been a major effort to collect data sets that can characterize, describe, and help us track the emergence and the spread of these diseases. So in fact, you can see that this information is becoming not only collected, but widely accessible. And so there are millions of sequences and hundreds of thousands of full genomes that are available on repositories that are hosted by the National Center for Biotechnology Information, NCBI, and many other databases, like Joint Genome Institute's integrated microbial genome databases. And these databases are growing in size at an exponential speed. And we recently saw that some of these are becoming more and more visible even to the public as we see how the call to apply bioinformatics tools to the analysis of such data are coming out. For example, in a recent review article in Nature, there has been a call to train more people to be able to utilize genomic data to help track and spread the disease, as we know, COVID-19 caused by SARS-CoV-2. So these kinds of data sets, because they're so widely available, you can see that it's easy to find and download this kind of data. It's easy to kind of develop a way to track the newly published data sets, but there's more to the story. In fact, in the same article on Nature that was published, I think a couple of days ago, it's been reported that even though we have a lot of information about confirmed cases coming out, the number of sequences varies drastically by country, and the quality of that sequencing is even further kind of a subject to investigation. So whenever we have this explosion of data and public awareness of the type of information that this data can produce, we also need to understand how this data can be properly analyzed and what kind of information can we get out of this type of data. So bioinformatics methods that find individual mutations, like multiple sequence alignment, can be combined with evolutionary models, which we need to understand, and track changes in the genomic sequences that we can link to a process. And these models fit to thousands of genomes, reveal the dynamic tree of the pandemic, which shows that the virus, for example, in the case of COVID-19, continues to change with novel distinct clades separating from a common origin over the course of the past year. And so now we know that there are approximately 12 distinct clades, and their number has grown. As you can see, there have been 3 clades identified just since November 2020. The clade proportions are likely to be unreliable, as I mentioned, because of the different levels of sequencing and the quality of sequencing done in different countries. But nevertheless, this information has become prominent and central because all of us are used to seeing this kind of information even in the media. So as we think about the dynamic character of viral infections and infectious diseases in general, we might ask the question, why, or what are the processes and kind of the reasons for such variation and dynamics over time? So what has recently been reported is that we now understand how pathogenesis, mechanisms of interaction between the viral and the pathogen and the host, the spread of resistance, and even immune responses can all play major roles in how we observe the genomic sequence changes in these kinds of data sets. So here you can see how different reports from kind of a time course of infection and different types of analyses from convalescent plasma have led to our understanding how these mutations evolve and how do they appear in the genomic sequences. And in fact, we now know that the individual viral genomes that we are typically thinking of a virus are actually whole communities or swarms of viruses that could be referred to as quasi-species. And so today, this is becoming an important part of diagnostic assays. So here you can see a publication that shows the representation of the hypothetical impact of comorbidity on SARS-CoV-2 quasi-species generation and testing, where wild-type SARS-CoV-2, or the most common form, is a reference for development of a diagnostic real-time PCR assay. So here you can see how this type of information could lead to a much better understanding of comorbidities, inflammation, and increased rates in mutations that occur in specific populations. So as we develop novel techniques and a deeper understanding of what genomic data can produce, we also need to evaluate the methods of analysis that have developed tremendously over the past 20 to 30 years. And today, we can even look beyond individual mutations. For example, another recent article, it talks about multinucleotide deletions, where whole regions are being deleted from these genomes and how those impact specific clades in the phylogenetic tree, and even phenotype, like the ability to cause persistent infection and more severe infection. So as a result, what we have done is we have tried to introduce a number of such important tools for genomic data analysis into a user-friendly T-Bioinfo platform that allows anyone, biologists, clinicians, as well as bioinformaticians that have to deal with hundreds or even thousands of samples at a time, to analyze this type of data efficiently. On our platform, we provide specially designed sections to deal with infectious disease and other type of data. So in the context of this genomics program, what we want to do is go through all the different types of analysis methods that are available so that we can try to apply them to specific types of infectious outbreaks that are relevant for this cohort. So we'll talk again about sequence alignment and mapping, how to detect variants based on multiple sequence alignment, or look for variants and their proportions in next generation sequencing FASTQ files. We'll also talk about analysis that allows us to look at frequency of mutations, as well as call variants with specific level of confidence, and use that to find differential mutations, or mutations that can differentiate between different groups of samples in our data. We'll also talk about the process of annotating those mutations and understanding the impact of the mutations on the amino acid or the protein sequences. We'll look at a number of different types of pipelines that could be built using standardly used tools with some modification, and we'll look for also advanced analyses like copy number variation. We'll also see how genomic data matched with numeric data about phenotypes can help us detect specific variants that are responsible, and combinations of variants, in fact, that are responsible for phenotypes such as resistance to specific drugs or vaccines. We'll also go beyond the scenes and go into how we can create reports using R and Python, loading the final sequence alignment and producing detailed reports on specific positions, and also on how these positions could be used to understand the variation in the data and how to select the specific types of mutations that are most commonly reported and used. For example, the variants of concern in the case of SARS-CoV-2. And so the two major directions for this upcoming genomics program is to talk about malaria and COVID-19, or SARS-CoV-2. So for malaria, we'll speak about different aspects of the importance of understanding of the overall infection cycle and how that infection cycle can help us put a context on the genomic data with different tools like sequence alignment, as well as visualization and working with 3D structures of proteins. So in this program, you will see how we will cover a number of specific types of analyses, starting from the very basics, going on into user-friendly analysis tools and project examples, and then incorporate some of the training in coding in Python and R, so that most of you that join this program are enabled and are experienced enough to be able to design and develop your own genomics research projects related to infectious diseases. So this is a program that we've done before on different types of diseases like cancer and others, but here we will focus specifically on the application of genomics to infectious diseases. Now, some of you joining today might not have a lot of background in genomics, so we will provide you with information starting from the very basics. So we'll talk about the basic terminology, what are the building blocks of DNA, and various types of DNA variation like synonymous and non-synonymous mutations, what is frequency of variants, how do we differentiate between single nucleotide polymorphisms or SNPs, and copy number variation. You'll become knowledgeable how to use these terms appropriately to describe different types of variants and master several examples of their importance in various conditions. We'll also discuss the process of DNA sequencing and the different types of DNA sequences that you might find online. And we'll see how the analysis of whole genome and exome sequencing can be used to find variants and study association between genotype and phenotype. We'll also introduce you to self-guided exemplar projects that will talk about different types of infections that are important and relevant to genomic data. For example, we'll talk about Ebola virus, the outbreak in 2014, 2016, how to find specific genomic genomes in cell lines that are used for biologics. We'll talk about the origins and pathogenesis of the novel coronavirus, and we'll see how to use genome-wide association studies to understand tuberculosis infection and drug resistance. So when we talk about Ebola, it's a great example because it was one of the first outbreaks that we saw almost real-time generation and sharing of large-scale genomic data. In this project, you will see how the tracking of the infection can be applied to genomic data that is available on NCBI virus, and you will be able to create phylogenetic trees to study genomic association with geolocation as well as time. You'll also see how mapping of those mutations between nucleotide and amino acid sequence reveals important variation in groups of samples that are available on these public repositories. But we'll also go beyond that, and we'll talk about this intra-host variation where you can see local haplotype reconstruction using methods that analyze closely related sequences in raw reads of FASTQ files. As a result, you will be able to use this information to track Ebola virus transmission and see the impact of specific variants on vaccine design. In another project on the origin and pathogenesis of the novel coronavirus, we will also talk about how we can detect where was the first emergence of this important pathogen and how we can use phylogenetic analysis to track the development of this pandemic and try to see how closely related this novel pathogen is to some of the previously studied and well-characterized coronaviruses. Again, we'll use multiple sequence alignment where we compare different sequences from SARS, bats, pangolin, and other types of coronaviruses to understand the impact of specific genomic variations on protein structure and interaction. But we'll also talk about the emergence of treatment approaches to this important pathogen, and we'll see how transcriptomic data can also be used to find association between treatment response and specific types of pathways that are responsible for efficacy and inflammation. So ultimately, you will find that the application of genomic data will be described for viral genomics and evolution, for immune response and biomarkers, and microbiome and infection. And so we will lead from genomic data characterization into multi-omics integration, ultimately linking the genomic variants with pathway analysis. In another example for tuberculosis, we'll talk about genome-wide association studies, where our goal is to find the association between specific individual variants and the different types of resistance to either a single type of treatment or multi-drug resistance where multiple types of treatments are being used. So in that case, you will learn how to use genome-wide association studies and use different approaches for GWAS analysis to ideally understand the specific genes and their mutations in the context of many different samples that we will analyze together. And so we will also look at how to produce a report and characterize the resistance emergence throughout different regions around the world. Now, as I mentioned, genomic data characterization has improved to the level where we can go beyond individual mutations and start looking at other types of outbreaks where defective genomes are responsible for spread and disease process. So here we will use the enterovirus D68, which was reported across the different parts of the globe, especially in the US and Europe. And we will see an outbreak of this virus in 2012, 2014, and 2016, where there was a different pathogenicity between different populations. So here we will use a phylogenetic tree reconstruction algorithm to find out whether EVD68 strains from different outbreaks at different time points are similar or different based on the phylogenetic analysis of patient data. Here we will use the variation and the strains to identify clades related to specific dates of outbreaks. And we will finally look at how specific variants can characterize these individual strains that are either on the nucleotide or the amino acid level. So to talk about how you can gain access to a lot of these program resources, I will pass this back on to Bipsa, and she will introduce you to the process of registration.
Thank you so much, Ilya. So I will continue by sharing my screen right here. So as I'm going to be talking a little bit about the program resources, I want to get started by launching a poll here on the screen. So this is just a poll to know more about who you are. As you see that in the chat, we have participants who are coming from different countries around the world. They have different interests. So please let us know. The first question here, as you see, is which country you are from. So there are several options. If you're from any other country, then you can mention the other countries that you're from in the chat box. The second question is, what is your level of education? Then the third is, how familiar are you with genomics? As we are talking primarily about genomics, so we are going to be asking you the question, but in case you're also interested in any other topic, like Anirudh mentioned, more about NGS data analysis focused on transcriptomics or microbiome data analysis. So if you're interested in any of these topics, you can also let us know in the chat box. And if you share your email ID with us, so we will be reaching out to you. And the last question I wanted to add here was, how did you learn about the genomics webinar? So where did you come to know about this webinar that you're attending today? Did you receive an email from us? Did you see the webinar on Facebook, LinkedIn, Instagram, or was it recommended to you by any friend or peer or faculty? Please put them down in the poll. So we'll keep the poll on for just another minute while I answer a question from Polomi in the chat box. So Polomi says, I'm an undergraduate biotechnology student. Can I do any of the courses you're providing? Yes, Polomi, you can do any of the courses we are providing. And this is because of the program resources that I'm going to be sharing. So you will notice that when we are talking about the resources, it starts with courses which begin at a very primary and introductory level. So we give you access to these resources from where you can start learning and supplement that learning along with online sessions like this webinar that we are having today. So as part of the Omics Logic Genomics program, we will be having online sessions. I will be quickly taking everybody through that schedule shortly and also show you how you will be learning from each of these resources. So every week we'll give you some assignments, give you some tasks that you have to complete. And this you can do along with any of the other studies that you're doing. If you're a college student or if you are pursuing your PhD, you can do this. So if you are looking forward to join a program like this, we recommend a minimum of 6 to 8 hours per week if you want to make the best out of the program. And even if you are someone who is very busy, may not be able to attend all of the online sessions, we also record each of the session videos and send it to our participants. So the session recordings will also be available for all of the sessions that we will be doing. Okay, so I'm reading some of the questions that are here. So I will also be introducing everybody to the other programs that are available. as part of the Omics logic programs that we do, and we have several of those. But today, as we are focused on genomics and infectious diseases, so we are going to be talking a little bit about this and moving into the other programs. And everybody who is sharing, please continue to post your email IDs or any of the feedback that you have in the chat box. Today's session, Tanvi, is free. Of course, a training program is not free of cost. So I will be talking about the costs right at the end. So I think we have received the poll. Ilya, Dr. Mohit, can you confirm? Because I cannot see if the poll is being done. So I can end the poll now.
Yes, I think you can end the poll. I see there's been 75% voted.
Okay, thank you so much, Ilya. So I will be ending the poll now. Thank you all for sharing. So Omics Logic program is built to provide students of all backgrounds with a clear perspective on big data, analytics, machine learning, and artificial intelligence in application to omics data for research, clinical, and industry applications. Our Omics Logic step-by-step process is enabled by access to complex tools via a simple and intuitive interface, at the same time exposing participants to analysis logic in the context of real data sets and extrapolation of this logic to a variety of projects and data sets with assistance from mentors and technical support, as we have seen Ilya tell us so far. The program licenses include access to tutorials and project examples, big data analysis tools, coding training, and mentor support. So this is all included in all of the program licenses, and I'm going to be talking in depth about this in short while. So let us start by signing up to EduTBioInfo. So if you are not already a member of the platform, please start by signing up to EduTBioInfo. And Dr. Mohit, if you can kindly share the link in the chat box. The EduTBioInfo portal has introductory courses, which leads to multiple other courses and projects that can serve as a springboard into the world of bioinformatics. And here learning about genomic data analysis. And we also have several projects that you see, some of which Ilya has also discussed. For the duration of the program, we will also be using the TBioInfo platform to process big data sets. This platform is built for multi-omics integration and analysis of structured data with advanced methods like machine learning and mathematical modeling. For downstream analysis and visualization of processed data, there are many packages in popular coding languages like R and Python, which you can learn on the Code Omics Logic Playground. Additional coding exercises will also be used to practice sequence alignment, evolutionary studies, and interpretation of found variants. So let us now go together to see how to register for this program. So for this, I will be sharing my screen. And on my screen, you have the registration page link. Thank you all for sharing the email ID right now. So this is the registration link. So now let's quickly take a look at what we can see in this page. So we have the start date, which is 12th of March. So we are going to be starting very soon, in less than 10 days. The program time is the same time that we started today, which is 9 a.m. CST or 9 a.m. Central Standard Time. And if you are from any other country outside of the US, please convert it to your local time. In India time, it will be 8:30 p.m. IST. In GMT, it will be 3 p.m. GMT. And once you go down here, we have also prepared a video for the session that you can see, so you can learn more about what we are going to be talking about. Here is a program overview. And once you scroll down, you find the schedule for the session. So here is the entire schedule for the session. So you can actually go through the different topics that we will be dealing with in each of these sessions. And we will have bi-weekly sessions here. And let me take you through the registration process. So once you're in this page, if you're looking forward to get started, you have to click on Get Started, and you will notice that we offer 3 licensing options. So this is what I was referring to when I meant that we offer licensing for this program. So these licenses cover the resources that we have just spoken about earlier. So we have a beginner's license. So what does the beginner license constitute of? The beginner license constitutes of all the resources we mentioned, which is the asynchronous course materials, the live sessions, the hands-on assignments, group sessions, as well as projects. And how is it different from the intermediate and advanced? So the intermediate comes with an extra benefit where you can request time for one of the mentors to mentor you on a one-on-one session. So if you have any questions, or if you feel like that you want to take the program at a pace as well as learn from one-on-one sessions and not just the group sessions, you can request for this, and for that you can take the intermediate license. While the advanced license is a research license, which means that if you are looking forward to doing research using genomics data or similar kind of data, then what we recommend is having the advanced license. So what does the advanced license give you? It gives you an additional 30 days. So we already see that the program is going to be ending on 15th April. So for you, if you're taking the advanced license, it's going to be giving you additional 30 days. So which means that your program access will end on the 15th of May. And you can actually do a research project in that extra period of time along with our mentor team, and also work together with the research fellows we have. And thank you so much, Bhagat Singh, for sharing. So these are the different options we have for payment, and we will be sharing any other details that you require. And we also understand that there are some students who might need help in joining the session. So we also offer scholarship. So I will also paste the scholarship link right in the chat box. And the goal of the scholarship is to offer students who are bright and who might need help in attending a training program like this. And there are some requirements that a student would need to follow to fill up and to get access to the program as part of the scholarship. So if you noticed on this page, you have a form. Now, what you need to do is, if you're applying for the scholarship, please make sure that you're filling up all of these fields. So if you just write nil, your scholarship will not be granted. So we need to especially meet with you and your reference. So this is very important. The goal is to make sure that you are going to be following through a program like this, that in the faith that we are providing you the scholarship, you are going to be doing the tasks that are assigned to you. So basically, if you are receiving a scholarship, you will be assigned few tasks, which means that you have to give us constant updates of your progress throughout the training. You have to send us reviews of the courses that you do. So you have to be very involved in this entire training. So once you're filling out the form, please make sure you do this as soon as possible, because we will have to set up meetings with you and one of your references, who can be from the institute or the university that you come from. So we also present to them this opportunity, we talk with you, and then based on that discussion, you are offered the scholarship. So if you want to take scholarship for this program, please mention here as well which program you're applying for, and anybody can apply for this scholarship, but it will only be given once we have that discussion with you. So Bhagat, can you see the chat box? I've shared the scholarship link again, and I will also share the link for the genomics program right here on the chat box here again. And while I'm talking about this, I also wanted to invite everybody to join our social media groups. So in the social media groups, you will see that we have a lot of participants who are coming from different parts of the world, and they are just like you, scholars, students, researchers, as well as faculties and administrators from different parts of the world. So it's a great place for you to go ahead and get started knowing from different people and collaborating with us. So we believe in collaboration, and we do hope that we are able to collaborate with you. And we also have a Facebook page. So I'm also sharing the link here of the Facebook page. And let me paste it in the chat box. So these are the few pages that we have for different sections, and whichever social media you are comfortable using. And we also have other programs that are coming up. So as Anirudh was requesting, we do have this transcriptomics program that we started on 2nd March. So we have had only one session so far. The next session is going to be on Friday, which is March 5th. So Anirudh, if you're interested in learning more about NGS data analysis, and not only Anirudh, anybody else who is interested in learning more about this kind of data analysis, you can go ahead and check out this link. The registration process is the same, as well as the scholarship link remains the same. Just when you're applying for the scholarship, please mention the program that you're interested in, as we have several different programs that we are running at the moment. So I do hope that you all are able to take part in programs like this and take this opportunity. And I'll quickly stop sharing and find the link to the other programs for everybody here. So, okay, this is the link to the other programs that we are having as part of the several Omics Logic programs that we are doing this spring summer. So let me quickly again share my screen and share this link. So via this link, what you can do is see the different programs that we have. So we have a research fellowship program. Shubham, we do not offer any PhD programs, but we do have a program on research fellowship, and I will quickly explain what it is. The goal of this program is to help you design or develop your own novel research project. So this is a program completely focused on research projects. It's offered for either 3 months or 6 months. And we work with students, we work with even high school students who are doing really well under this program, where they're getting started on their research journey way earlier. And that is possible using the resources that we offer, like the big data analysis server platform that you saw, which has multi-omics integration. So you can understand the different applications and how easy it is, because it's there in the cloud and a student just needs to log into the platform in the browser and use the platform. So this is the Bioinformatics Research Fellowship Program. And the next program that we have is this program that we are seeing today, which is the Omics Logic Genomic Program. So whenever you are applying for any of these programs, please make sure that you're selecting the program you're interested in. And this is the Omics Logic training program that we are running currently in India, but we also accept any kind of international participation and whoever is joining us now. And we also have another program coming up focused on precision oncology. So this program is going to be starting on March 15th. And this is also another program where we are going to be focusing more on cancer biology, tumor development, etc. And if you are someone who is working with cancer, or you're interested in going towards a research area like this, then you can also apply for this program. And I'll just show you how to apply for this. So you notice that there is a form at the end of the page, and you can actually choose from this different programs that you would want to apply for. So once you select a program, you will receive an email regarding the program. So please make sure that you're selecting the program of your choice. And if you want to apply for multiple programs, then please fill up the form twice and make sure you're selecting the options, different options at this different time periods. And thank you so much. If you all have any questions, so research fellowship, right? So, okay. So Manisha, what do you want to know more about the research fellowship? If you can let me know, then maybe I can clarify it for you. While for metagenomics, yes, we just concluded a metagenomic program. So we had this metagenomic program that we started in February. So it's going to be expiring very soon. So for all the programs, so all the programs have different costs. So you would have to go ahead and subscribe and see what all these programs cost for. So maybe you can reach out to me, Ayobami, and I can share with you if you want to join all of these programs. And please note that some of the sessions may be clashing. So we would recommend you to start with one and then progress to others, because we'll keep on repeating these kinds of programs even in the coming summer months. So this is my email ID. Thank you, Dr. Mohit. marketing@pine.bio is my email ID for reaching out to me.
Yeah, thank you, Beepsha, and for everyone reaching out for us for these different programs that we are offering. So this is the website where you can actually apply for these programs, and then you will get emails about the entire process, that how we are going to conduct the sessions, when the program is going to start, and all the other details like the free sessions that we are doing associated with these programs. And also for research fellowship, we are doing this with participants who have who have learned from one of the Omics Logic programs, and now they want to work on their project and finalize a project and then move to a way where they can present the project and show it to their peers, and also like maybe publish. So that's the entire structure of the research fellowship program. However, I mean, if you're interested, please go to this link and fill up the form, and we'll be emailing you as well. Thank you.
Yeah, thank you so much, Dr. Mohit. And I would just want to answer some of the questions which I see right now. So when exactly is the research fellowship starting? So the research fellowship does not have a commencement date. Our research fellows work throughout the year on different research projects. What we aim to do is help you develop your own research project. So you can start joining today, and you will get started with your 3-month or 6 months research journey. So the research fellowship, again, to answer Swati's question, will it be containing details on data science applications with healthcare? So each of the research fellowship program is focused on training you in an area you are interested in and developing a project. So mainly if you're interested in working on a project, we start with the training and help you go through this. And Ilya will also be explaining the process shortly. And to answer Avosise's question, is the application for research fellowship free, or does scholarship cover it as well? So the application again for the research fellowship program is not free, but definitely you can again go ahead and apply for scholarship. The criterion of selection for scholarship, as I mentioned, is based on the conversation that we will have with you on a one-on-one meeting with your mentor. So please make sure that the details that you're entering is correct, and it will also require you to perform certain tasks, as I mentioned, which we will mention to you during the meeting that we have. So now I would like to pass back this session to Ilya to discuss some of the projects. Over to you, Ilya.
Okay, thank you, Bipsa. Again, thank you everyone for the informative questions. I briefly wanted to mention that many of the programs that you are seeing here are designed to help you conduct independent research. And to do research using bioinformatics, it's important to find a data set, to learn about the application of different methods. And it's also a collaborative process. So you really need to work with others on different components of your project. For example, if you come from a bioinformatics background already, you might want to find a collaborator that is more biology versed. If you are coming from a biology background, maybe you will find it challenging to conduct certain types of research with coding and computational tools, and also describing your methods. So the program that we have, and we typically have between 10 and 20 fellows at a time, we try to facilitate this process allowing you to gain access to all of the different training sessions that we have, know about the different types of analysis using different tutorials and project examples. And we typically see a slow transition between learning to then designing your research question and finding the data set that you will study that question using, and then eventually working with you to help you polish up the presentation and potentially even write a paper. So I pasted a link here to a number of recent projects that have been completed by different participants. And I want to just show you, this is just a few examples, really. It's not a comprehensive list, but to get you an idea, one of the major kind of stepping stones for a lot of people once they design a project is to be able to get some feedback on what it is like. What did people think about it? What do other people who are either experts in this field or experts in bioinformatics, what can they say about the project? And so typically that has been done in the context of poster presentations where students can present their posters and get some feedback, answer questions, etc. So we essentially provide an online version of that by conducting regular meetings where both experts and other peers can provide feedback to a project presentation, and also eventually when the project is done, and sometimes that project is ready for a publication, we guide you on how to submit that publication and help you make sure that the draft is good. And then also we have a number of collaborators at the Tauber Bioinformatics Research Center and in other places that we ask to evaluate the project from an expert perspective. And so here are some examples of posters that range from Alzheimer's disease to cancer to infectious diseases and others. Some people take very cutting edge projects like the effect of radiation on the microbiome in space, and other people apply it to very immediate types of challenges. You can see here, for example, identification and imaging of potential vaccine targets for SARS-CoV-2 spike. So really diverse range of projects that are either done independently, like here you see a project by URJA, and sometimes in groups. Here you can see a collaboration between a number of different people that work together. So oftentimes, I think as a result of such collaborations, you get to understand a project from multiple perspectives. And it also, in the context of a short time frame, can prepare you for a longer plan to conduct a project in a more thorough way. So for example, our bioinformatics for infectious disease program with the University of Ghana is scheduled for 6 months. And the reason for that is because just now, after maybe a month and a half of training, the participants are starting to really work on their project presentations. And that's one of the things that I actually wanted to mention, that we have an upcoming session where you can see some of those presentations, where students and faculty and researchers that have completed some training program will be sharing their project proposals. We will have this available on March 10th. It will be a session where we will have some of our research fellows present some of the people that have worked under a scholarship that have summarized kind of what they have learned. And we will have some of the participants of the first phase of the Bioinformatics for Infectious Disease program, where you will find proposals on malaria and on SARS-CoV-2. So Beeps, I don't know if you can share the link for this session, or if you have submitted your email address, we will be emailing you that information maybe by the end of this week. So we welcome those of you, especially those of you that are interested in applying bioinformatics to different research problems, to reach out to us. Maybe some of you would like to speak about your project and about your, what to do with your project, essentially. So please do reach out to us. Dr. Mohit Mazumdar here is curating a lot of data sets and helping groups of students kind of develop their project proposals. So you can definitely reach out to us, marketing@pine.bio or mohit@pine.bio, to schedule a discussion about your project. We also want to mention that in many cases, the way this works best is for faculty that do not have sufficient time to dedicate to a project, but would like help from their students being guided by some expert to develop an analysis. So another option is really to join a program together with your students, maybe 2 or 3 students that can do the analysis, and you can kind of provide the oversight and guidance on selecting the topic and making sure that the analysis is good. So if you have any questions about those, please do reach out to us specifically, and I'll pass it on back to you, Beepsa, to kind of answer some of the questions that are being asked.
Thank you so much, Ilya, and thank you, Manasa, for sharing with us this feedback. I hope that everybody is now able to see my screen. So at this point, I would like to ask, if you have any other questions, please feel free to reach out to us. So we have shared 3 email IDs in the chat box. One is mohit@pine.bio to reach out to Dr. Mohit regarding any kind of questions that you may have regarding projects, collaborations. If you want to reach out to us regarding the research fellowship, the programs, or any other opportunities, please feel free to reach out to me, marketing@pine.bio, even regarding any kind of access that you would want to different websites that we have or the different portals that we offer. And we also have a team in Africa based out of Nigeria. So you can reach out to our Africa team if you're based out in Nigeria, Africa, or Ghana, or any of these countries. Africa@pine.bio is the email ID. So if you do not have any other questions, thank you all for joining today's session. So Abukari, you can reach out to Aditayo. So Aditayo Borisade is a community manager in Africa. So regarding any kind of problems with credit card or any other payment issues for participants in Africa, Nigeria, please reach out to Aditayo. So africa@pine.bio is the email ID. You can also send that email to me, and I will be able to help you connect with Aditayo as well. So thank you, Aditayo, for sharing this. So do we have any other questions? And thank you, Polomi, for sharing the feedback. Thank you, Burhan. Thank you, Ehsan. Okay, yeah, so Delago, yes, we do have professionals as well who apply for this program, which means that professionals who belong to any of these domains in biotechnology, bioinformatics, and they too would want to learn more about bioinformatics application, especially industry professionals. So yes, we do also train them. So if you have any specific requests regarding that, please reach out to me. I will be able to help you. So again, I would like to invite everybody to connect with us. So here you would be able to see the different people who are there in the groups, interact with them. We do hope that you're able to interact with us and ask us questions as well. We are also active on Instagram. So please go ahead and follow us on these links. On Instagram, you can find us at Bioinformatics Pine. And we are also there on YouTube. So our YouTube channel name is Pine Biotech. As you saw that Dr. Mohit had shared earlier, we had streamed this session live on YouTube. So it will be available there for review if you want to go ahead and review this later. So thank you all. I would want to end today's session. I look forward to seeing you all in the upcoming Omics Logic Genomic program or any of the other programs that we are offering. Have a wonderful day, everyone. And please feel free to email us regarding any questions that you may have. Thank you and have a good day.